Hello, my name is Seungjun Ahn from the University of Florida. As a quick overview, we have slides for the background, the pseudo-value approach, simulation, and results of the simulation and the real data application. The differential network analysis, the DN analysis, to be short, identifies connectivity changes among set of genes in a network, but there is no available direct regression modeling for DN analysis that regresses the gene expression level between the two comparing groups, such as smokers versus non-smokers, with the inclusion of additional clinical covariates, such as patient age. So here we propose PRANA, which is a pseudo-value regression approach for network analysis that can adjust for additional covariates. The main idea of this slide is that we focus on the total connectivity, which is one of the characteristics of a network, which is simply the column sums of the association matrix using the RNA-seq data, which is denoted as the data hat or the data K hat, which is the total connectivity for gene K. And similarly, we go ahead and compute the data K parentheses I hat, which is the column sum of a gene K calculated from the re-estimated association matrix using the same RNA-seq data, but now without the ith subject. So based on data K hat and data K parentheses I hat, we can then compute the data tilde, which is the jackknife pseudo-values. So next, we fit a robust regression to regress the data tildes on a set of covariates, which can be expressed as the following equation. In our study, the t-test was used to test whether the true difference in total connectivity of kth gene differs between the two comparing groups. And the empirical Bayes screening approach is applied to control the false discovery rate. The simulation. So we picked 20, 50, and 100 genes to test PRANA from smaller to larger gene networks. And for each gene network, the following sample size specifications were considered, and we ran our simulation 1,000 times. Precision, recall, and F1 scores were assessed to evaluate our model performances. So here's a network plot. Well, actually, the network plot with covariate dependent structure, which visualizes the scenario settings of our simulation, where age acts as a confounder. That is an observed difference in connectivity, maybe due to a difference in the distribution of age. So here's the simulation results. So by and large, the PRANA, our proposed method, resulted in a higher recall than DNA path, and also had a higher precision compared with the Dingo. And generally, the PRANA maintained a high precision recall as well as the F1 scores, regardless of the gene and the sample size specifications. And a mild note for Dingo, which is one of the competing methods. So large size specifications were not feasible for the simulation due to heavy computational costs and computational time. Application study. So we have analyzed 28 COPD-related genes with 406 sample sizes from the COPD gene study, which you can download from the GEO database. So for PRANA, DNA path, and Dingo, so for all methods, the binary current smoking status variable was used as the grouping variable. But for our method, PRANA, the following set of variables were also included as additional covariates in the multivariable model. So as a result, 23 out of 28 COPD-related genes were predicted to be differentially connected between the two smoking status variables, while accounting for the set of covariates. And of the 23 DC genes from prana, 5 are found additionally. And of these, we have found that a gene called DMWD and THRA were related to rare genetic outcomes. And here's a slide for the Venn diagram to summarize the analysis of the real data, the COPD gene data. So conclusion, prana reaches a consistently high degree of precision and recall to identify DC genes with varying simulation sizes. Future studies are warranted to extend our work to other measures of association of a network, which assess different structural changes in the network. So here's a slide for the acknowledgement for my funding and the references. Thank you.